Okay, listen carefully. At the end of this lecture, even though class is supposed to officially end at 9:20, I'm going to hang around, but I'm not going to record it, but I'm going to hang around. I had one person send me an email about some questions about Lab 4, but I want to start by saying this first and foremost, okay? I don't give much feedback because that feedback for most of y'all would involve giving you the answer. These are labs we have to use for future semesters, and we are staying online for the spring, just to give you a heads up. Okay, that's already set in stone. Um, so therefore, we don't give the answers back. Okay, main reason, we've had a significant amount of trouble with cheating. I mean, it's been through the roof. Okay. Apparently, we have a lot of students that feel that attending ACC, and I hear it's the same thing at the four-year colleges. They're having problems too. Apparently, many students see college as an extension of K through 12, and they refuse to grow up. But I will stay. I will give you some feedback. You're not going to like most of it because it simply has to do with, hey, look, you need to read the lab. You need to study the lab. The answers are right there. Okay. But I will go over it some at the very end. All right, let's continue. Let's get it going with chapter 6. I want to finish chapter 6 today, chapter 7 on Thursday, and then you have an exam from a week from today. It'll be 50 questions, multiple choice, same format. But let me make something clear now about that too. There are no excuses. Okay, you need to make sure your internet connection is correct or find an ACC campus to show up to and use their Wi-Fi. Okay, we had one problem with one student out of 4 classes. Everybody else was fine. Excuses won't fly. Make sure you take care of everything, please, okay? So again, I had one problem with one student over 4 classes. If you add in the summer, that's 5 classes. There shouldn't be any issues, okay? So make sure, because that's on your shoulders there, okay? Enough with all that. Let's get going here. Chapter 6 focuses on one of 3 types of energy producing processes called cellular respiration, okay? Cellular respiration, the purpose is to produce ATP, okay? And this is performed by cells of our body, eukaryotic cells, more generally speaking. So most of this process occurs in the mitochondria. And the energy that is produced, okay, is coming from food or nutrients, specifically the nutrient glucose. The nutrient glucose also serves as what we call chemical energy. Talked about this before. There are those living organisms that have the ability to synthesize their own nutrients. Some of that nutrient serves as chemical energy. We call them producers, such as the plants in the background. Other organisms are what we call heterotrophs. They do not have the ability to synthesize their own nutrients. Therefore, these living organisms, called consumers, must consume other living organisms to obtain their nutrients, and some of that nutrients will serve as chemical energy, which they're showing you here. Well, of the energy producing processes, 2 of them are shown here, photosynthesis and cellular respiration. We cover cellular respiration in chapter 6. We'll cover photosynthesis in chapter 7. I put these 2 on the same slide because you can see there's a relationship between the 2. Okay, they refer to them sometimes as 2 processes that create a cycle of energy production. Okay, these 2 formulas you're going to have to be able to recognize on the test. Again, it'll be multiple choice, so be able to recognize them. Photosynthesis requires 6 molecules of CO2, 6 of water, photons, which are light particles. Now, the purpose of photosynthesis is not to synthesize ATP. The purpose of photosynthesis is to synthesize the nutrient glucose. But we end up with the waste product. It's a gas, O2. Okay? Photosynthesis consists of a series of chemical reactions. None of the chemical reactions require O2 to produce the product. Therefore, anytime you have a chemical reaction or a group or series of chemical reactions catalyzed by enzymes that do not require O2 to help produce a product, we refer to those chemical reactions or those series or groups of chemical reactions as anaerobic metabolism. Remember, metabolism is chemical reactions in a living organism, catalyzed by enzymes. This chapter, however, focuses on cellular respiration. Cellular respiration. The purpose of cellular respiration is to produce ATP. In order to produce ATP, a cell needs a nutrient. Okay, the nutrient can be a protein or a lipid too, but the preferred nutrient is carbohydrate, specifically glucose. Okay, glucose is not only a nutrient, it serves as a type of chemical energy that a cell can take advantage of and use to help it produce ATP. Now, in order to do this, okay, it requires a group of enzymes, many are actually three groups of enzymes that carry out three different groups of a series of chemical reactions. And along the way, as these chemical reactions are being carried out, O2 is needed. So overall, the process cellular respiration involves, okay, at least one chemical reaction that requires O2. Therefore, cellular respiration is referred to as aerobic metabolism, okay? In order for a cell to produce the amount of ATP it needs from one molecule of glucose, in order to not only survive but to function, O2 has to be present. If O2 is not present, say it's skeletal muscles of our body, if O2 is not present, skeletal muscles of our body or organisms such as yeast can perform fermentation. The problem is, which is anaerobic metabolism, and ATP is produced. Fermentation is a third of three types of energy producing processes. Now, fermentation has that in common with photosynthesis. They're both types of anaerobic metabolism. However, fermentation has something in common with cellular respiration. The purpose of fermentation is to produce ATP. And there's two types, alcohol produced by yeast and lactic acid by your skeletal muscles. The problem is not enough ATP is produced, okay, to keep the cell alive through this process. So when it comes to ATP production, cellular respiration has to occur. O2 is needed. You can see the cycle here. Well, what this cycle is basically showing you is that the products produced by photosynthesis happen to be the reactants needed to perform cellular respiration. And then if you look at cellular respiration, two of the three products produced by cellular respiration are needed for photosynthesis. So what I'm getting at is if you know the formula for one, you already know most of the formula for the other. You just got to flip it. The difference is going to be for photosynthesis, you got to add in photons as one of three reactants. For cellular respiration, you got to add in ATP as one of three products. Now, as I said, what we're going to focus on, almost hit the wrong key there. What we're going to focus on is cellular respiration, this process here. So listen carefully. For cellular respiration, these two right here are all reactants. As I said, cellular respiration, as we're going to talk about, is divided into three stages. Each stage consists of a series of chemical reactions catalyzed by enzymes. In order for a chemical reaction to occur, we need reactants. However, if an enzyme is catalyzing a chemical reaction, at least one of the reactants, which in this case is glucose, at least one of the reactants has to actually be a substrate. Remember, all substrates are reactants, but not all reactants are substrates. The substrate is the reactant that an enzyme will directly bind to in order to produce a product. Now, does that make oxygen not as important as glucose? Actually, they're both equally important. As I said, a cell will not produce the amount of ATP I'm showing you here unless O2 is present. If we were to remove O2 from this cellular respiration formula, only the three stages would occur, and we would only get 2 molecules of ATP. The cell wouldn't even survive. It would die. Okay? So O2 here, it does have a very important role, but it's not a substrate. I'm going to draw a little line here. The role O2 plays, the way it functions, is that O2 functions as the final electron acceptor. I'm almost there. Okay, it functions as the final electron acceptor. I even talk about insecticides, antifungal creams, as well as two poisons, cyanide and carbon monoxide, that interfere with O2 functioning as a final electron acceptor, thus cutting off the majority of ATP production, and the cell dies. Therefore, the organism dies. So glucose is the substrate, O2 is the final electron acceptor. Okay. Now, for cellular respiration, it consists of 3 stages. As I go over this, the purpose of the first 2 stages is to strip as many electrons from glucose as is possible. Well, you have to keep in mind, okay, and if you need to, put see chapter 3, carbohydrates, and chapter 2, covalent bonds. Glucose is an organic compound. Therefore, what atom forms the backbone of organic compounds? Everybody, there's 11 of you here. What atom forms the backbone of organic compounds? Right, carbon. And what type of chemical bond does carbon like to form? You only have 3 choices. Right, covalent. And what is a covalent bond? What's the simplest definition? If I just never mind the number and the polarity, what's the simplest definition for a covalent bond? It involves the what between, right? Electrons are shared. Therefore, as I go over the first 2 of 3 stages of cellular respiration, the focus is to strip electrons from glucose to take them to the 3rd stage. Okay? So that must mean when you take electrons from an organic compound, you're probably breaking what? The covalent bonds, not just between the carbon atoms that form the backbone, but also the hydrogen and oxygen atoms that contribute to the formation of the organic compound. So you're going to see when I show you the first 2 stages, it's going to say the first stage focus is the breakdown of glucose. The second stage is to complete the process of breaking down glucose. But what's actually happening is electrons are being stripped. Now, is there a little ATP produced in the first 2 stages? Yeah, and I'll show you this. But the main reason is to strip electrons from glucose to take them to the 3rd stage, because it's the stage, the last stage, where most of the ATP is produced. Okay, and this is where O2 comes in. The electrons oxygen accepts as the final electron acceptor, they actually came from who? Glucose, the substrate. Okay, so I'm saying this once, but I'm going to go into detail. Okay, I just try to get repetition in because several years back I had a student tell me the number one thing they needed was to hear it again and again and again, and I said that's understandable. So we breathe. So I'm a little out of order in your notes right now, but basically what I'm covering is the page 1 and the very top of page 2 in your notes. So respiration, simply breathing. Everybody knows you need to inhale O2 and exhale CO2. Everybody knows that if you can't breathe, you can't get in O2, you're going to die. But what nobody knows is the reason why, right here. The reason why you need to constantly inhale is, and inhale specifically O2, atmospheric oxygen. Okay, it's because cells of your body need it to perform cellular respiration. The only cells that are not performing this energy producing process called cellular respiration are the ones that are dead. Okay, but therefore, therefore cellular respiration is continuous. It's nonstop. The only thing that changes for cellular respiration is whether a cell's being told speed up the rate at which you're producing ATP or slow down the rate at which you're producing ATP. In other words, increase cellular respiration or decrease, but it's always happening. Well, not only do you get ATP as a product during the cellular respiration, but you get water and you get CO2. So you always need O2 because of cellular respiration, but it's also because of this process, you're always producing a waste product called CO2, so you have to exhale it. This is what's going on with the 2 gases here. As I said, there's fermentation. The only thing I want you to know for fermentation is it's 1 of 3 energy producing processes. It falls under anaerobic metabolism. It does not require oxygen, but it does consist of multiple chemical reactions catalyzed by enzymes. The purpose is the other thing I want you to know. Purpose of fermentation, produce ATP. Okay. The last thing I want you to know about fermentation is there's 2 types. Alcohol fermentation, such as carried out by yeast, and then the other type of fermentation, lactic acid fermentation or lactate fermentation performed by your skeletal muscles. Our skeletal muscle cells, when you start exercising, rely on cellular respiration to produce enough ATP to ensure that your muscle cells can contract and produce movement. But if you exercise to the point of fatigue, eventually your skeletal muscles from cellular respiration to lactic acid fermentation. Okay? The problem is we get much less ATP produced, therefore your skeletal muscles really don't last much longer. Okay? And what happens is when you exercise to the point of fatigue, your skeletal muscle cells, which are now using this type of energy producing process, lactic acid fermentation, to meet the demands you're placing on them, eventually your skeletal muscles say, that's it, we're not responding anymore. We are actually going into survival mode. We have just enough ATP to stay alive. We're not going to do anything you want. I don't know if any of you ever gone to the gym and you got carried away lifting weights, especially like bench press, and you think you can do one more and the mind's willing, but your arms aren't responding, your chest isn't responding, and the bar's slowly coming back down on your chest, and you start realizing you should have asked for a spotter. I don't know if any of y'all had that happen. Or I have a son that lives near the Rocky Mountains. You ever go hiking up into the mountains? Not the hills like we have in Texas, but the mountains. I don't know if you ever done that. And you think you can make it all the way up, and all of a sudden your legs just stop responding. Okay? All right, for cellular respiration, there are 3 stages. 2 of the 3 stages happen here in the mitochondria, stages 2 and 3. Okay? So I'm going to cover a little bit of the anatomy of the mitochondria, and then I'm going to revisit this towards the end of this chapter. The mitochondria is a membranous organelle. It's an enclosed structure that has 2 membranes, an outer membrane and an inner membrane. And these membranes are made up of the same compounds that make up our cell membrane. Okay? So both of these are phospholipid bilayers, and in particular, the inner membrane was found to contain many transmembrane proteins that function as enzymes, and not just enzymes, but enzymes involved in cellular respiration. 2 of the 3 stages, the second and third stage of the cellular respiration, will happen here in the mitochondria across this inner membrane, catalyzed by these enzymes. So if you look over here, it says organic molecules and O2 have to be transported into the mitochondria so these enzymes and proteins can do their jobs. This is where the majority of ATP is produced, but we also get our waste product being produced here also, CO2. Okay, so as I keep saying, cellular respiration is broken up into 3 stages. Collectively, all 3 stages together are referred to as aerobic metabolism. So therefore, cellular respiration falls under aerobic metabolism. Cellular respiration has to do with harvesting of energy from glucose to produce ATP. And it takes 3 stages. The 3 stages are glycolysis, the citric acid cycle, and oxidative phosphorylation. Make sure you know the names of the stages, everybody. Okay? Know that glycolysis is stage 1. Know that the citric acid cycle is stage 2. And know that oxidative phosphorylation, okay, is stage 3. If you look here, glycolysis focuses on the breakdown of glucose into 2 molecules of pyruvate. I'll show you a figure that's the best figure I've been able to come across to simplify what's going on. What I don't say here is that what's causing the breakdown of glucose, well, I said it earlier in the lecture, electrons are being stripped from this organic compound. The second stage, called the citric acid cycle, it completes the breakdown of glucose, or what I call glucose-derived products, molecules that have come from the substrate glucose. An example, pyruvate. It's the final product produced at the end of glycolysis. Where did this molecule come from? Glucose. Okay, so we'll talk about specifically a substrate called acetyl-CoA, and this molecule is derived from the breakdown of glucose. And we'll talk about how when this molecule, the substrate acetyl-CoA, enters the citric acid cycle, okay, enzymes that catalyze a series of chemical reactions in this cycle complete the breakdown of this glucose-derived product by stripping even more electrons. So the purpose of these first 2 is to strip electrons. And not just strip electrons from glucose and glucose-derived products, but then to transport those electrons to stage 3. Okay, Stage 3 is called oxidative phosphorylation, and it's actually broken up into two substages, which I didn't remember to number in your notes, but they are bold type towards the end of your notes. So oxidative phosphorylation is the third stage, and it's broken up into two substages. The first substage is simply called electron transport. And we're going to talk about how there's a network of proteins that perform this process, but they require not ATP. They require the flow of electrons through them to give them the energy they need to do their job, called electron transport. When that group of enzymes does their job, called a process called electron transport, that will then allow an enzyme known as ATP synthase to perform the second substage of oxidative phosphorylation, called chemiosmosis. So there are many, many enzymes. Sorry, this should be a C, which I'm making worse. You ever do that like that? Get carried away with making it better and it looks uglier. Okay, so listen carefully. Each of these three stages consist of a series or a group of chemical reactions catalyzed by a group of enzymes. Okay, but there's really only one enzyme I'm going to want you to know, and it's going to be here for chemiosmosis. Okay, it's an enzyme called ATP synthase, and it synthesizes the majority of ATP. We'll visit that when we get there. I'm not going to test you on this table, but it's just showing you glucose isn't the only nutrient that can be used as a type of chemical energy to produce ATP. Okay, fatty acids can be used, as well as the molecule they're attached to, glycerol. Proteins, specifically amino acids, can be used. Okay, but our cells prefer not polysaccharides, but this should say monosaccharides, such as glucose. Glucose being the number one nutrient, not just carbohydrate, but the number one nutrient that our cells prefer to use. Because when our cells can use the carbohydrate glucose, the monosaccharide glucose, glucose will be used for the first two stages. Therefore, glucose is used to catalyze all three stages. This stage should say oxidative phosphorylation. If you look here for, say, fatty acids and amino acids, they could either go through all three stages, in the case of amino acids, or only through two stages. But what they're not showing you is that over 10, 15, 20 years, if the cells of your body are not able to use the sugar, specifically glucose, okay, as the substrate for cellular respiration, if instead over 10, 15, 20, 30 years, your cells have to use fats and proteins as the nutrient to produce ATP, the waste products produced aren't simply CO2. And this is what wreaks havoc on a person that's diabetic, okay, over long term. You might have heard somebody with type 1 diabetes, or maybe you or somebody in your family has it. Type 1 diabetes, which is sometimes still called juvenile diabetes, although they're going to have to start reclassifying everything. That's a type of diabetes caused by a person's own immune system attacking their own cells that produce insulin. Therefore, basically, once a person's born, slowly over time, their cells become more dependent on needing this. But that causes problems with the body. So the person I met that had type 1 diabetes to where their body couldn't take advantage of using glucose and instead had to use this was a 38-year-old man who had his one leg from the knee down amputated, on the other leg from the ankle down amputated. Okay, you end up with a lot of serious side effects. So we're going to focus on the nutrient glucose for this process. That is preferred. Listen carefully. Of the three stages, stage 1 glycolysis occurs in the cytosol. So make sure you know that. It occurs in the cytosol. Stages 2 and 3, the citric acid cycle and oxidative phosphorylation, they occur in the mitochondria. Okay. When we go over stage 1, glycolysis in the cytosol, it begins with the substrate glucose. It consists of a series of chemical reactions catalyzed by a group of enzymes, and the final product produced is pyruvate, but not just pyruvate. It'll be 2 molecules of pyruvate. Then we'll talk about how those 2 molecules of pyruvate are transported into the mitochondria, and an intermediate step happens. On page 2, under glycolysis, the last bulletin is an arrow. Okay, this intermediate step is not part of stage 1 or 2. It's just an in-between step. And we will talk about how each molecule of pyruvate is converted into a molecule of acetyl-CoA, so we'll end up with 2 acetyl-CoA. The reason for this is because the first enzyme that catalyzes the series or group of chemical reactions that make up the citric acid cycle, that first enzyme that gets the citric acid cycle started doesn't recognize pyruvate as a substrate. It recognizes acetyl-CoA. Then we'll talk about how the citric acid cycle, we don't talk about a final product. There is no final product. Okay? Acetyl-CoA and pyruvate are examples of glucose-derived products. Notice the arrows are letting you know where they initially came from. As the acetyl-CoA enters the citric acid cycle, many, many more electrons are stripped from it to where we have basically nothing left. Now, if you look at stages 1 and 2, we do get a little ATP produced, but they're showing the little symbol here to indicate most of it's produced here. Now, I would talk about this as we go. The electrons that are stripped from glucose and the glucose-derived products, pyruvate and acetyl-CoA, those electrons are transferred to stage 3 by molecules called electron carriers. So I will go over these guys that they have written abbreviated, NADH and FADH2. As I said, the 3 stages are catalyzed by a series of enzymes. Many of the enzymes have in their names kinases. Okay, so for my majors class, they actually have to know a handful of enzymes, such as for glycolysis. They have to know the first enzyme that acts on glucose, and they have to know the last enzyme that produces pyruvate. And both of them have in their name kinase. Okay, a kinase is an enzyme that phosphorylates molecules. Okay, so a kinase is an enzyme that phosphorylates molecules. What do I mean by phosphorylates? Phosphorylates is when a phosphate group, okay, is covalently bonded to a molecule, such as when this enzyme wants to produce ATP, it's a kinase, it will phosphorylate ADP. It'll take a phosphate group, chemically bonded to ADP, producing ATP. So if you look here, glycolysis, this is in your notes, okay? You may have to add in a little extra as we go. This is glucose. If you notice, there are 6 charcoal colored beads. Well, that's because the molecular formula of glucose is C6H12. I want to screw this up. I screwed it up yesterday for my majors. O6. All monosaccharides, glucose, there you go, I screwed it up at the end, talked too soon. C6H12, this should say O6. All monosaccharides, such as glucose and fructose, have the same molecular formula. However, the structure formed is not always the same. Okay, fructose is what we call a pentagon. It has 5 sides. Glucose is a hexagon, what we call 6 sides. But what they show you here is representing the 6 atoms of carbon that are covalently bonded to each other, forming the backbone of glucose. When glucose enters glycolysis as a substrate, the enzymes that act on glucose to strip electrons, some of these enzymes require ATP. Well, if you exercise, you know you have to expend energy in hopes of having more energy later. It's the same thing for a cell. The cell's going to have to use some of the ATP, but the trade-off is it's going to produce a lot more than it uses. So it's going to have a net gain. The cell's going to end up ahead. So when glucose, the substrate, enters glycolysis, the enzymes that act on it to strip electrons, some of them are going to need some ATP, total of 2 molecules for each molecule of glucose that enters glycolysis. Adenosine diphosphate, we covered that in chapter 5, part 2, okay? Is it better? Just go back to chapter 5, part 2. But adenosine diphosphate, 2 phosphate groups. So go back to chapter 5, part 2, towards the beginning, I gave you the structure of ATP. I don't know if you remember or if you looked at it. I gave you the structure of ATP. all you would do is remove one of those phosphate groups with phosphorus and oxygen and just leave two. That's ADP, adenosine diphosphate. That answer your question? Okay. So as glucose enters, some of the enzymes need ATP to help them strip electrons from glucose. And what ends up happening, well, glucose is an organic compound. There are a lot of covalent bonds involving the sharing of electrons, not just between the atoms of carbon forming the backbone, but even the hydrogen and oxygens. But in particular, the carbon backbone, as electrons are being stripped from glucose, that carbon backbone is broken. It goes from a 6-carbon backbone, one 6-carbon backbone molecule called glucose, to 2 3-carbon backbone molecules. If you notice here, they're trying to show you something happens to these molecules. Okay? Listen carefully. This right here, both of these, is referring to a molecule called an electron carrier. There's a total of 2 different types, and you're going to have to write this down. I don't give you all everything in your notes because I'm already giving you all enough. And sometimes I feel like that backfires. These are molecules called electron carriers. Okay? Now the way we write these molecules, and I'm going to circle these down here. The way we write these molecules is that if the molecule called an electron carrier, if this one of 2 types is written this way, NAD plus, positive, that means there are no electrons. What do I mean by that? That means it's not carrying any electrons. Okay, so there are no electrons. However, as they're trying to show you up here, these electron carriers, okay, can actually carry up to 2 electrons. Why are they putting a bead with the little minus sign on it, everybody? Why do they use a bead with the minus sign to represent an electron? Anybody know why? Y'all should know. They have a negative charge, exactly. So they're not showing you the symbol E with the superscript negative. They're showing you a little bead, not with the minus, but with the negative. In biochem, what they do is if the carrier's not carried, if the electron carrier is not carrying electrons, what they will do is they will write it down as NAD+. Now, everybody, the NAD is an acronym, okay? This electron carrier has a very long name. The N stands for nicotinamide, just to give you an idea. Okay, we don't need to know the name, the full name. Okay, it's kind of like those chelators, but if you want it, I'll send it to you. Okay, so what a chemist does is if they want you to know this molecule called an electron carrier is not carrying any electrons, they will write it NAD+. That no, it's not carrying any electrons. But what happens is these electron carriers, such as NAD+, will come into these first 2 stages and an intermediate step. And electrons stripped from glucose will be given to these electron carriers. Thus, notice this electron carrier comes in and it gains 2 electrons. It picks up 2 electrons that came from glucose. Another electron carrier comes in and it gains or picks up 2 electrons that came from glucose. They are going to transport them to stage 3. So how does a biochemist let you know when the electron carrier is carrying 2 electrons? They'll rewrite it as NADH. So when you see it written this way, this is not only telling you it's an electron carrier, but it's telling you it's carrying electrons. Okay? These electrons by the electron carrier, let me erase all this. These electrons that have been picked up by the electron carrier, now written as NADH, they're going to take the electrons to stage 3. Oxidative phosphorylation, and not just any part of stage 3. As we'll talk about, they're going to take it to the first substage, which is electron transport. Okay. Now, if you notice, after the electrons have been stripped from these glucose-derived molecules that have a 3-carbon backbone, also some ATP is made. Okay. We have a total of 4 molecules of ATP. So over here, I'm going to put a minus. The cell has to use 2 molecules of ATP, so it's, in a manner of speaking, it's subtracting 2. But over here, at the end of glycolysis, or towards the end, the cell actually produces 4. So what's the net gain, everybody? Does the cell come out ahead in the end? Yeah. It ends up ahead by 2 molecules of ATP. After that happens, we end up with our final product, 2 molecules of pyruvate. Pyruvate is a glucose-derived product. Each molecule of pyruvate has a 3-carbon backbone. Where did these atoms of carbon come from? Glucose. What caused the backbone of glucose to be broken? It was tied into the electrons being stripped from it. Okay, so the final products then overall is our net gain of 2 ATP, our 2 pyruvate, and our electron carrier NADH transporting electrons. That's what we have at the end of glycolysis. Okay, the electron taken to stage 3, the cell will use the ATP. Maybe it'll put the ATP back into glycolysis to produce another 4. Who knows? And then the 2 pyruvates, okay, will be transported into the mitochondria. So here where it says organic molecules, what it's actually referring to is pyruvate. So listen to me carefully, everybody. This figure here that I'm circling with the little finger, and this figure here that I'm circling with the little finger, they are the same. They are the same, okay? I have students that they look at this one and it makes much better sense, and I have students that look at this one and they say it makes better sense. In either case, I'm going to go up and down. Okay, so we have 2 molecules of pyruvate. If you go down here, the pyruvate produced at the end of glycolysis will then be transported to the mitochondria. Mitochondria is single. If we take away the O and put an A, mitochondria are plural. Either one's fine. What will happen is when pyruvate is transported into the mitochondria, which they show you down here, but they don't show you here, what will happen to the pyruvate, okay, is that an enzyme complex will come in and strip electrons, breaking off one of the 3 carbon atoms. So here's our 3 carbon. Here it's giving you more detail. When the electrons are broken, that 1 carbon atom that is now released happens to be attached to 2 oxygen atoms. Therefore, it's released as CO2. So notice here, less detail. Here, a lot more detail. The electrons that are stripped are given to the electron carrier NAD+, so we rewrite it as NADH to let you know it carried 2 electrons. If this doesn't make sense, come up here. This is the same thing. Here comes NAD+, electrons from breaking off that 3rd carbon. Now it's going to carry these electrons to stage 3. So this is one way of showing it. This is another way of showing it. Okay. Now what happens is that same enzyme complex that has performed these first 2 processes, resulting in the release of CO2 and the electron carrier NADH transporting 2 electrons to stage 3, this same enzyme complex will grab an enzyme called coenzyme A, and it will covalently bond it to the remaining 2 carbons that were left. What was once pyruvate, once that first carbon is broken off, it's acetic acid. Once we add this molecule called CoA, now we have our final product, acetyl-CoA. This has more detail than this. Okay, so this is the intermediate step. In your notes, to the left of that arrow, write down intermediate step, and that it is not part of stage 1 or 2. It's an in-between step. It begins with pyruvate. It ends with the product acetyl-CoA. And you have CO2 produced. And you have NADH transporting 2 electrons to stage 3. This is what happens in the intermediate step. We begin with the substrate pyruvate in the intermediate step. We end up with the product acetyl-CoA. We also get some CO2 being produced. And we end up with the electron carrier NADH transporting 2 electrons to stage 3. Now, what's the purpose of this intermediate step? this right here, acetyl-CoA. Okay. In order to begin stage 2, the citric acid cycle, okay, the very first enzyme that catalyzes the group or series of chemical reactions, that very first enzyme that starts off the citric cycle, it doesn't recognize pyruvate as a substrate. It recognizes acetyl-CoA. So first of all, listen carefully. This slide here, as well as slide 15, they're the same. It's just two different ways of representing it. Okay? So whichever one is more pleasing to your eyes. I'm going to try to use this one mainly. Okay? So what happens is a citric acid cycle, like glycolysis. Okay? It consists of a group or a series of chemical reactions catalyzed by a group of enzymes. The very first enzyme that gets a citric acid cycle started, it doesn't recognize pyruvate as a substrate. It recognizes acetyl-CoA. Okay? So I'm going to use this other figure here. So acetyl-CoA will enter the citric acid cycle. And as acetyl-CoA enters the citric acid cycle, the enzymes that catalyze this series of chemical reactions, which they have numbered, these enzymes are focused on stripping even more electrons from this glucose-derived product, or another way to say it, completing the breakdown of this glucose-derived product. And how are they completing the breakdown? Notice here, there are 4 different points at which they show electrons being picked up by electron carriers, except this one here. I already talked about NADH as an electron carrier, but there is a second electron carrier. So these are both electron carriers, except this second electron carrier, you only see it during stage 2, the citric acid cycle. Make sure you make a note. That's the only time you see this second electron carrier. This second electron carrier, if it is not carrying electrons, we write it as FAD. If it is carrying electrons, we write it as FADH2. Now remember, the test is a multiple choice test, so you just have to be able to recognize the molecule, its abbreviated name, as well as that it comes in 2 abbreviations. Okay? So if you look at these 2 electron carriers, go back one. That's why I gave you this slide. Here's the electron carrier, NAD+, when it's not carrying electrons, and here it is when it's carrying electrons. So it'll come in, and at 3 different points during the series of chemical reactions, 3 of these guys will pick up 2 electrons each, and then take them out of the citric acid cycle and take them to stage 3. But now we have a new electron carrier, and one of these will enter the citric acid cycle. For every 1 molecule of acetyl-CoA that enters this second type of electron carrier, only one of them will enter the citric acid cycle, but it will also pick up or carry 2 electrons out of the cycle and also carry them to stage 3. So anytime an electron carrier gains electrons or picks up electrons, it's always taking them to stage 3. Now, where these electron carriers play a role? Well, NADH, it's carrying electrons to stage 1, stage 2, and the intermediate step. But this electron carrier, FADH2, only carries electrons out of stage 2 to stage 3. So this is over here showing it to you here. Now, using this other figure, as acetyl-CoA goes through and it's being stripped of more electrons, we get CO2 being produced for every 1 molecule of acetyl-CoA that goes in, and we get 1 molecule of ATP being produced for every 1 molecule of acetyl-CoA that's going in. But keep in mind, okay, okay, that for every 1 molecule of glucose, we actually get 2 pyruvate. So both pyruvates are being converted to acetyl-CoA. The reason why I'm saying that is because we get a net gain of 2 ATP here. And since from 1 molecule of glucose, we end up with 2 molecules of acetyl-CoA entering the citric acid cycle. And for each molecule of acetyl-CoA that enters the citric acid cycle, we get 1 molecule of ATP. We get a total of 2. In other words, the formula I gave you for 1 molecule of glucose, we should get approximately 36 molecules of ATP. Well, you have to account for it's not 1 acetyl-CoA from 1 molecule of glucose, it's 2. That way, the number of ATP produced add up close to the 36. So by the time the first 2 stages are completed, we have a net gain of 4 ATP produced. Up here it's showing you, it's missing here. There should be one here, but you can see the electrons being carried out of these stages and the intermediate step are all being transported to oxidative phosphorylation. Vivian, are you trying to say something? I don't know what a slash means. Oh, okay. Fell asleep? You fell asleep, didn't you? Okay. All right, so listen carefully. Okay. So by the time the citric acid cycle is completed, we don't talk about a product like we do glycolysis or this intermediate step. Okay. The glucose-derived molecule acetyl-CoA that entered this cycle, so many more electrons have been stripped from it. There's nothing left. There's no product. Okay. But when the second stage is completed, now enough electrons have been stripped from glucose and glucose-derived products to help catalyze, okay, or to help promote the first substage of oxidative phosphorylation called electron transport. Listen carefully, everybody. This line right here that I'm pointing at with the arrow, that's the inner membrane of the mitochondria. Okay? Over here on this side, this is the outer membrane of the mitochondria. If we were to magnify either of these membranes, you would think you're looking at the cell membrane. And that's what I'm going to show you. The figure I'm going to show you has magnified just the inner membrane. That's this here. So this structure right here is known as the inner membrane. So I'm just going to put IM for inner membrane. And if you recall at the beginning of the lecture, I said the inner membrane has many enzymes or proteins inserted into it. Okay. Now going back to this figure here. Here's our inner membrane. Here's our outer membrane. If you look, the two membranes are not connected. There's a space in between them. So this gap that I'm running the little finger along, that is called the intermembrane space. On the other side, okay, enclosed within the inner membrane, on the other side, enclosed within the inner membrane, or surrounded by the inner membrane, this area here is called the matrix. So another way to say it is this is the very inside of the mitochondria. So notice something. Here's the intermembrane space. So what we can't see is over here would be the outer membrane. And then we said the space enclosed by the membrane, the space enclosed by the inner membrane. We call that the matrix. Just to give you an idea of where we are. All right, so listen carefully. This third stage is called oxidative phosphorylation. It's broken up into 2 substages. One is called electron transport, the other is called chemiosmosis. These 2 substages, the names of these 2 substages are processes, functions, catalyzed by proteins. Okay, and most of these proteins are enzymes, or what we call enzymes. First substage of oxidative phosphorylation. Well, the oxidative, the first part of this third stage name, comes from this here. Y'all don't have to know this, but my biology majors that take 1406 do have to know this. Whenever a molecule such as an electron carrier gains or picks up electrons, we say it's reduced. However, whenever a molecule such as an electron carrier loses electrons, okay, or drops them off, we say it's oxidized. That's where this is coming from. It's what happens during electron transport. If you look here, here's our electron carriers, and notice the abbreviations they're using. NADH and FADH2. What does that mean? That means that they are carrying electrons. It says from fruit, but it's from glucose. What happens is NADH that has left stage 1, the intermediate step in stage 2, carrying electrons, NADH will take them to stage 3, and specifically, it will take them to a protein complex called the electron transport chain. So all of these proteins here, all of these proteins that I'm trying to circle here, make up the electron transport chain. Sometimes I write out the name in a multiple choice question. Sometimes I use ETC, so know both. Okay. This network of proteins that form the electron transport chain, they don't get their energy from ATP. They get their energy from electrons flowing through them. That's what the yellow lines mean. So the NADH molecules that transfer electrons from stage 1, the intermediate step in stage 2, they will bring their electrons to stage 3, specifically to this protein complex called ETC, and they will let go of their electrons. They will lose them, and the electrons will flow through this network of proteins called ETC. At the same time, the other electron carrier that leaves only stage 2, FADH2, it will transport its electrons to this third stage, the first substage. It'll transfer its electrons to this network of proteins called the ETC, and notice the yellow line means it's letting go or losing its electrons. So these electron carriers transfer their electrons specifically to the ETC, which performs the first process of oxidative phosphorylation called electron transport. As the electrons flow through the ETC, this gives the ETC the energy it needs to pump protons out of the matrix and into the intermembrane space. This in turn creates a proton concentration gradient. Well, if you don't know what I mean by concentration gradient, again, go back to chapter 5, part 1. Also, today's lab focuses on membrane transport. It'll also mention concentration gradients. Everybody, I have a lot of students, because they're non-majors, they think this course should be a pushover. Well, that depends. I literally had a high school student tell me that the way I teach 1408 was easier than her high school class. Okay, but there was about 5 students standing behind them that didn't agree with that. So it's a college level course, okay? So you need to go over chapter 5, part 1 to help you understand electron transport and chemiosmosis, in other words, to understand oxidative phosphorylation, but also to help you with today's lab, which focuses on membrane transport. So as electrons from these electron carriers are released and flow through this network of proteins called the electron transport chain, that gives the ETC the energy it needs to function as a proton pump. They will pump protons, H pluses, out of the matrix into the intermembrane space, creating a proton concentration gradient. We have an area of high proton concentrations outside of the inner membrane and an area of low proton concentration within the inner membrane. Okay? Now, the electrons that are flowing through the ETC eventually are accepted by our final electron acceptor. Ignore all this other stuff here. Okay? Eventually, the electrons that flow through our ETC exit the ETC, where our final electron acceptor, O2, is waiting to accept the electrons. And what happens when O2 accepts the electrons? It's converted to H2O. Okay? Now, O2 has to be present to accept electrons. O2 has to be the final electron acceptor. I'll show you how there are poisons as well as chemicals used that block this process and cause the death of an organism. But when it's functioning properly and O2 is here to function as the final electron acceptor, we end up with the proton concentration gradient that now causes the second substage to occur, chemiosmosis. So electron transport is required to promote chemiosmosis. Chemiosmosis is defined as work performed in a proton concentration gradient. Okay, so chemiosmosis is defined as work performed in a proton concentration gradient. Well, again, we have our high proton concentration out here. And I'm saying chemiosmosis is defined as work performed in a proton concentration gradient. Well, what does the work in cells, everybody? What's the answer to that in general? What did I say does the work in a cell? Not a rhetorical question. What does the work in a cell? It's one of four organic compounds, right? Proteins, okay? Are enzymes proteins, yes or no? Are enzymes proteins? Yes, okay, so listen carefully. So when I say chemiosmosis is defined as work performed in the presence of a proton concentration gradient, well, I'm indirectly telling you there is a protein that's doing this work, and it's not any protein. If it's an enzyme, what three letters did we say it'll end in? Often, not always. ASE. Okay. What does the work in the presence of a proton concentration gradient? It's an enzyme called ATP synthase. Okay. The reason why the ETC needs to create this proton concentration gradient is so that the protons will want to diffuse from a high concentration to a what? Low. But the protons cannot get through this phospholipid bilayer. However, look at ATP synthase. In its name is what it does. It synthesizes the majority of ATP produced during cellular respiration. Okay? But it needs energy to make this ATP. So where does it derive its energy from? Notice, it's like a channel protein. It actually has two functions. Okay? The first function is it actually allows protons to diffuse through it from a high concentration. They will diffuse through ATP synthase to a low concentration. But as these protons are diffusing from a high to a low concentration through this enzyme, that's giving the enzyme what, everybody, that it needs to make ATP? That's giving the enzyme the energy it needs to synthesize ATP. ATP is the primary source of energy for many proteins, everything from membrane transport to enzymes, okay? But it's not the only source. And again, comparing it to vehicles, most vehicles, I believe today, still run on gasoline, but there are those that run on diesel, there are those like Teslas that are electric, and there are those that are hybrid, okay? So there's a primary source of energy, but that doesn't mean there aren't other sources of energy, okay? That's the same case here. So ATP synthase performs chemiosmosis. The type of work it does, it synthesizes ATP in the presence of a proton concentration gradient. But it takes advantage of that proton concentration gradient. It allows protons to diffuse through it from a high to a low concentration, thus giving it the energy it needs to do its work, synthesize the vast majority of ATP. But what happens when protons diffuse from a high to a low concentration? In order to prevent these two solutions in the inner membrane space in the matrix, in order to prevent these two solutions from reaching equilibrium in regards to proton concentration, the ETC is kicking them back out. So these protons are just going like a revolving door, back and forth. This keeps this third stage or this second substage, chemiosmosis, that keeps the enzyme continuously producing ATP. When did I say that a cell is not performing cellular respiration? When's the only time a cell isn't performing it? What did I tell y'all? Yeah, when it's dead. That's literally true. So if you look here, this one's two more than what I gave you at the beginning. So on the test, just as long as you see somewhere in the 30s, 30 to 38, you know I'm giving you the formula, okay? But notice, the purpose of glycolysis is not to produce ATP, but we do get a net gain of 2. But the purpose is to strip electrons, take them to stage 3. We have our intermediate step where more electrons are stripped, and then we have stage 2, the citric acid cycle. We get a little bit of ATP. We go from one glucose molecule to two, this says pyruvic acid, but it's pyruvate. Transported to the mitochondria, we get 2-acetyl-CoA derived from one molecule of glucose. Each molecule of acetyl-CoA that's derived from glucose enters the citric acid cycle, and we get 1 ATP for both. So we get a total of what, everybody? 2. In other words, we are tracking how much ATP is made from 1 molecule of glucose. So you can't say just 1 ATP produced for 1 acetyl-CoA. You have to remember, if we're tracking 1 glucose, we get 2 acetyl-CoA, therefore we get a total of 2 ATP. But that's only 4. The formula I gave you at the beginning said approximately 36. The squiggly line means, I forgot to tell y'all. The little squiggly line at the beginning in front of the 36 ATP, that's approximate. Here they're giving you 38. All I can tell you is in textbooks, I've always seen for cellular respiration somewhere between 30 and 38 ATPs produced. Depends on the cell. But notice something. The vast majority of ATP that is produced during cellular respiration is produced during the 3rd stage, specifically chemiosmosis, 34. So I use a squiggly line. Can y'all hear me? Have y'all been able to hear me? Okay, I'm sorry. My computer over here is already telling me it's messing up on my end. Okay, so this squiggly line means approximately or about. So notice, vast majority. And I said the key, part of the key or a major key is O2 has to be there as a final electron acceptor. To prove my point, poisons in cellular respiration. It's known that the gas carbon monoxide and the chemical cyanide, okay, they're both deadly to living organisms. And you hear it a lot in relation to us, okay? Well, the way they kill off a living organism, a eukaryotic living organism, is by disrupting cellular respiration. Cyanide and carbon monoxide actually interfere with the first stage, electron transport. It interferes with electrons flowing out of the electron transport chain to O2. Cyanide and carbon monoxide actually get in the way of this. So what happens is the organism is not able to produce not only enough ATP to do its normal functions, but now the cell can't even produce enough ATP to simply what? If the O2 can't do its job as a final electron acceptor, now the organism can't produce enough ATP to even survive. Well, there's also an insecticide called rotenone, which also interferes with O2 functioning as a final electron acceptor. And then there's an antifungal cream, topical cream that you rub on your skin, called oligomycin, and the way it kills off the fungus is by interfering with cellular respiration. It blocks O2 from functioning as the final electron acceptor. Okay, so therefore, what happens? This enzyme ATP synthase is responsible for synthesizing the majority, the vast majority of the ATP. Can't do it. So now the cell can't even produce enough ATP to stay alive. Thus, it kills the organism. Okay. So very general information, what I want you to know for these 4. Just know that the mechanism by which it kills organisms, it interferes with cellular respiration, specifically stage 3 oxidative phosphorylation. It interferes with the first substage, electron transport. It blocks O2 from being the final electron acceptor. Therefore, the cell can't even produce enough ATP to survive. Okay. Are there any questions on chapter 6 before I transition over to the lab here? Okay, I'm going to stop the recording on this.